Um, so the next thing that we have is just to open everything up for questions. If you have, you can drop them in the chat, you can raise your hand, and you can direct them either at all the speakers, or if you have questions specifically for one of the speakers, that's totally fine as well. Okay, maybe I will start off with just a question about some of the results that we saw. So Serge, in the results that you showed with the attention, and I know you said that it's not theoretically a shift, but necessarily just a multiplication between Gaussians. Did you find that your results depended substantially on the size of the original PRFs without the attentional field modulation? So in other words, do you see what appears to be a movement more if it's a smaller receptive field, let's say, or any kind of interaction like that?
Yeah, so that was the one of the initial results. So the change in the center of mass, or the shift, if you will, depends on the PRF size. And this is one of the sort of interesting findings that we found in the sort of regular resolution fMRI sort of results that sort of preceding these laminar results, is that we see that the changes in position are larger in later visual areas, and a lot of people would say, okay, well, that's in line with the effects of attention being bigger in later visual areas. But if we model it, we sort of kind of model them with the same attention field, and we can actually say, well, attention operates the same way across the visual hierarchy, but the effects of attention are depending on sort of the passive or the stimulus-driven sort of neural properties. At least in that data, it's a function of the PRF size, and not necessarily that there's differences in the way it operates in later visual areas. But this is, of course, a highly specific paradigm, right? So, but yeah, so basically it's a function of the PRF size. In the same way, if the attention field would change, that would also change the effects. But in the first study, we found the attention fields were actually very similar across the visual hierarchy.
That's really interesting. Thank you. It looks like, Camille, you have a question.
Yes, I have a question for Anna. Anna, that was a great talk, wonderful methodology, really impressive. However, I have a hard time with the result that the oxygen consumption is decreasing in layer 4. And my major concern comes from the fact that, if you look at tissues in general, and this is not just brain, but in many other tissues as well, there's a very strong correlation between tissue oxygen consumption, capillary density, and mitochondrial content. So when you look at that particular physiological data, that seems to go, contrary to your results. Do you have any comments on that?
Yeah, so this is a very good comment, and something that has been bugging me for a very long time. So this is also kind of why, to start with, my original hypothesis was that layer 4 should have higher metabolism. So maybe one thing to remember here is that we're looking at resting state. And so when Mother Nature puts mitochondria in somewhere, let's say in layer 4, it most likely means that under certain conditions, there is a massive energy demand in there comparing to other layers. However, we don't know when. So one possibility is that this has to do with presence of stimulus. The other possibility is that it has to do with certain brain states. So for example, maybe during the rapid eye movement sleep, there is a massive increase in oxygen consumption in layer 4. So the kind of the way that I now think of this is that I still believe that if we see that there is kind of like higher concentration of cytochrome oxidase and higher concentration of mitochondria in there, it's most likely there for a reason, but we don't know what this reason is and when it becomes physiologically kind of relevant.
do you think that you will follow through maybe by doing similar measurements under stimulus?
Yeah, absolutely. So we actually, as a matter of fact, as we speak, we're trying to do exactly this measurements. The problem that we have with this is that we don't have a very good model, which is not steady state. You know, so a steady state model is something that we can more or less stand behind because there are not too many parameters to fit. So on the other hand, when we have a dynamic model, then it becomes kind of, it's hard to underpin all those different parameters. So what we do is that we try to generate a pseudo-steady state where we just kind of drive it to a certain level of activity when there is an ongoing thalamic input. And we try to deliver this at frequencies where we confident that we don't, you know, like, depress kind of inputs or have some kind of saturation or something like that. So we're working on that. The other thing that we're trying to do is we combine this with those optically transparent electrode arrays that we have, and try to see how this oxygen consumption behaves as a function of the LFP power spectrum. So when we have more, delta or alpha or gamma, so what happens with oxygen consumption? And the third thing that we're trying to do is to look at this as a function of brain regions. Because some brain regions, like if you sit in primary somatosensory cortex, so there's, a lot of layer 4 going on at certain times. And when it's not going on, it's very quiet. But if we move to other cortices, we may see different pictures. So we currently are focusing on, prefrontal areas.
Thank you. Yeah.
Amir, you have your hand raised?
Yeah, very briefly to comment on this issue of oxygen consumption and layer 4. So indeed, if we look at cytochrome oxidase and also density of capillary and density of cells in layer 4, there is higher density mainly in primary sensory areas, such as V1, A1, and S1. But when you go to higher association areas, you don't see that. It's not in cytochrome oxidase that shows oxidative metabolism and not the density of cells and not the density of capillaries. So I agree with the hypothesis that probably needs still to be proven that this has to do with differences between external stimuli that are brought in through the thalamus and as opposed to resting state.
Yeah, just to add to this, so we are looking for that exactly reason. We're looking at this other higher associative regions. The other thing to keep in mind that also we measure, we believe currently, and this belief may be right or wrong. So we believe that if we estimate oxygen consumption around diving artery, so that's going to be representative for the entire cortical column. So there's some very good reasons to believe that it's can be treated as space invariant, within a layer, within a cortical column. But it remains a hypothesis. So we are working on some other ways to estimate this such that we're not going to be tied to that artery to be able to evaluate this hypothesis.
Bob, you have your hand raised.
Yes, well, I've put it on the chat as well. There's recent work by Micheva demonstrating that, at least in mouse brain, a lot of the horizontal interneurons that are myelinated are actually inhibitory. They're parvalbumin interneurons associated with basket cells. Now, I'm just wondering whether this fact might relate to Serge Dumoulin presentation. Alessio Fracasso's work isn't well known to me, because I used to talk to him a lot some time ago. Whether the sculpting of the population receptive fields across V1 and through V1 might be actually driven by activity in the stria genari. And the tightness of the receptive fields in V1, which is strikingly tight, might be actually associated with the strength of the sphere of Gennari. I just wondered if you had some comments on that.
Thanks, Bob. I think it's an interesting idea, actually. And so we did also in that paper look at the suppressive surrounds that we also measure with the PRFs. And we saw the similar kind of U-shaped function. But I think, yeah, the suppressive surrounds can be thought of as sharpening the PRFs, right? So I think, and this could well be mediated by those interneurons. So I think that is actually a very interesting thought. And so I think there's different ways to interpret this data. So our favorite way is sort of that there's a pooling going on from the central to the sort of deeper and superficial layers. But it could also be that there's more sort of inhibitory processes happening in sort of the central layers, which then sort of sharpen the response. I think that's actually a perfect valid interpretation given the data.
Okay, thanks. Well, I thought I'd get that in. I've been thinking about this for some time. There's some other very interesting findings about these parvalbumin and interneurons, which are coming out now in various labs, but I don't want to go into it now.
Yeah, so maybe sort of one comment in that the sort of the suppressive surrounds that we see sort of based upon negative bold, right? And they tend to be a lot larger than the typical ones that they see using neurophysiology. So there is potentially a discrepancy there. So they don't necessarily match one-to-one as a side comment.
Sure.
We also just had a question from Atina for Anna. So regarding the higher CMRO2 at layer 1, does it have anything to do with higher density of intracortical arteries at superficial layers?
Well, we don't think so. So the way that we estimate CMRO2 is not using the Fick's law. So we're not really, we're not kind of factoring flow into our estimation in any way. So we're just, it's a single kind of modality measurement. We just estimate CMRO2 based on PO2 itself. In terms of kind of how we measure this, in terms of kind of the underlying physiology of this, I don't think either, because I'm thinking of CMRO2 as something that has to do nothing with flow. So CMRO2 is basically just based on how much neurons are active and how much food they need. And the flow is happening in parallel. So in my view of the world, the CMRO2 and flow don't talk to each other.
Natalia? You're muted.
I was also trying to think about that topic. How exactly would it work then? What do you observe? And thank you for the interesting talks, actually, for all the speakers. But going back to that, so you do have a change in partial pressure of oxygen. You do have, you measure the CMRO2 based on that, but how do you get the oxygen there? So I think it relates a little bit to what Camille asked earlier. So is it the vessels are more permeable because you don't have the density to actually support it, right, in layer 1? So how do you think it happens?
I think what you're asking is how do we actually get kind of PO2? How do we achieve certain level of PO2 in layer 1? Like if the vascular density is low, how is it still oxygenated? So one thing is that arteries are hugely permeable to oxygen. And this was one of the first things that we discovered some, 12 years ago when we started doing these measurements with the first available probe at that time, that, people kind of used to think that just because their wall is so thick and there are multiple layers of cells in there, oxygen will be contained until it gets to capillaries, and that's where it can diffuse the tissue. So that is a very kind of wrong idea. It just happens to be that membranes and cells are completely transparent for oxygen. So oxygen is leaking from surface arteries. This also is probably the main reason why the periarterial spaces around the diving arteries are free of capillaries. It's just because this process of angiogenesis, you know, the biology of generating of vasculature, is sensitive to concentration of oxygen in tissue. And so because there's so much oxygen, capillaries basically are just not forming in there. Obviously there's some branches, right? But capillaries don't sort of like, you know, form around the arteries.
So if it's from the intracortical arteries, then doesn't that relate to what Athena just asked? Then I'm not quite sure. Because if you get it from the arteries, then it's just a different mechanism, basically, of getting the oxygen there. And I wonder.
Our estimation of CMRO2 has nothing to do with the kind of the mechanism that oxygen gets it. So oxygen is just diffusing. Oxygen is diffusing everywhere. It gets, you know, into the cells, it gets consumed, and so then there is more diffusion down the gradient. So there is no kind of like, it's, you know, it just has to do with the gradients. So oxygen and free diffusion.
Okay. All right. Thank you. I'll digest this. Thanks.
Write me an email.
Okay.
Perfect, Renzo.
Yeah, I have a question for Lars. Like you introduced this kind of development and learning at the beginning. So I was wondering if you think that this contextual processing is changing over the development.
Yeah, I guess in the sense that it greatly develops to early development, and then it's kind of set and fixed for later on. So I think the contextual in early sensory areas shouldn't change much. It picks up the image statistics and remains somewhat, and it's then a kind of different network that utilizes this kind of world models gained and learned to then use that in cognitive space to image comparison and so on. There's like some line of research where, you know, you do some image comparison shown in the periphery. You actually need the foveal representation in V1. If you TMS that, the precision of the comparison gets lost. Nancy Kenwish or Williams has done this line of research. So for this more abstract level, you utilize these models, but in a new way. That would be my interpretation, if that answers the question.
Thanks.
Luca?
Yeah, also for Lars, first of all, great data, beautiful data. It's beautiful to see some correspondence across species. I think that's really something that makes us believe into what we're doing, that what we're doing is true and real. But do I understand correctly? Maybe it's a two-part question. First of all, do I understand correctly? You're saying basically that you can identify two different types of like feedback, one to the outer layer, one to the inner layers. Would it be an oversimplification to say maybe the one in the inner layer would be more complex feedback, more high level? So while the outer layer may be a more simple feedback, so simple versus complex. And if that's true, do you think maybe we can, using training or using some specific tasks or different contexts, can we modulate the amount of feedback to go and directly test this hypothesis? So, you know, well, in this task, we would expect this and so forth.
It's early days and it's difficult, right? I mean, we make layer-fMRI look so easy, but easily, but it's a lot of work. So we are trying for years to do, you know, I mean, the one is like the navigation task or some cognitive comparison, or we combine tasks like the navigation. So you have your flat in your memory, and we look while you're moving around trajectories out of one room into the next room. How you process the expectation of the next room. We find some evidence at 3T, that's easy, but the layer-specific component is tricky. And then we also have the ambition to combine that with visual imagery. So you do visual imagery while you navigate. So the data so far is not yet, I mean, especially the last part, we don't have enough subjects, so it's not conclusive. The earlier part, we have some indication along these lines that maybe more for this complex combination, you do this in higher areas, and maybe more in deep layers, but it's still early days, I would say. We are excited about Johanna's data about the imagery versus the illusion, because we wrapped it up in one experiment, which we originally designed to kind of resolve other discrepancies we have with also Floris Lange's lab, who has seen, you know, amplitude effects in outer layers and inner layers in a bit of a different way. And so we use this decoding in the same experiment. We are confident about that because we replicated that across 2 different groups and variations and conceptually, but it's still early days. It also relies, so the mouse data that I showed, there is some mouse data for showing this learning, more complex learning, but that's somatosensory cortex. And there's also some evidence in auditory cortex for this association learning, but not yet from visual in the same. But otherwise, that's the idea, maybe more complex, counterfactual. So not this situation, but a different situation that you think about is processed in deep layers, independent of the superficial. But it's,
Anna?
So I have a question to both Lars and Serge. So when you guys are talking about this layer-specific results with both functional MRI, so how do you think about this? Are you thinking that this is due to flow, due to CMR2, due to both? And also, do you kind of just keeping in mind that if there is more CMR2 at certain place, so unless there is a oversupply of flow, that's going to contribute to negative bolt, not positive bolt.
Yeah, so I still have to digest the information you have shown. The way I've been thinking about it is it's energy driven, right? It's not oxygen driven. So and it's flow, it's volume driven. So from Camille's talks, Camille Uludag's talks, my interpretation was that at 7T we might be more able to tune into the volume changes, that are local enough to be picked up. Does that make sense? What's your interpretation?
Well, so I think that one thing that maybe, is different in a way that we approach this problem is that I'm pretty much convinced with all the measurements that we have done, and there's some very good work from others that basically shows that under normal physiological condition, it's not energy driven, so the vasodilation is not energy driven. So CMRO2 is energy driven because it just reflects energy demand. It's just as simple as that. But the flow, the vasodilation is driven feed forward from neurons. And, not even from astrocytes. So all the kind of the astrocyte, the metabolism, all that stuff, it becomes important as soon as we cross into pathophysiology, because those metabolites that have been shown to be vasoactive, they definitely are vasoactive. And astrocytes definitely can release vasoactive messengers. It's just that they're not the major driving force under normal conditions. As soon as we cross into pathology, they become the major driving force.
Serge, did you have anything to add to that, or?
No, not really. I mean, I think so. We're Yeah, I think there's many more people here who are more expert on sort of the neurophysiology than I am. So we're just using it towards cognitive paradigms. And I think that's why the comparisons to the neurophysiology is so crucial, to actually validate what we're measuring. But yeah.
Yeah, that makes sense. So we have time for a few more questions. Camille, you have your hand raised.
Yes, I wanted to maybe say something towards Anna's question about, of course, CMRO2 increases are actually will lead to negative effects on the BOLD. So will volume effects actually. Volume increases also in principle lead to negative changes in the BOLD effect. But the flow increases are the ones that lead to positive changes. And this at least not at the layer level, at least studies have always shown that flow increases dominate. They are much higher than CMRO2 changes, and so as a result you are getting the positive BOLD effect most of the time in areas stimulated, but of course there are negative BOLD effects as well. And so I would say that it would be very interesting to look at blood flow and CMRO2 and volume changes across the layers to answer Anna's question very rigorously. But in general, people report in many of their paradigms positive BOLD changes across the layers. So they are probably dominated by the flow changes.
Yeah, true. So one thing that kind of keeps me wondering is that both dilation and constriction propagate along the vasculature. Maybe not in the capillary, but definitely the arterial part of the tree. And they propagate, maybe like, 1 millimeter per second, something like this, maybe 10 millimeter per second. And so at the kind of scale of the columns, it's pretty fast. So the question is kind of like how, if we're not looking at the actual kind of times of onsets across the layers, but we're just looking at overall amplitudes, how would this, it kind of would wash out the differences between layers. That's kind of my intuitive thinking about this. One thing that we try to do right now is to just put like Matt Larcom, prism, like, put a prism in there so that we can see, look at kind of the diving artery throughout its length, just trying to understand what happens. But based on kind of current knowledge, it sort of looks like it should basically just like propagate throughout.
Of course, the kind of experiments that we've been hearing from Serge and and Lars, they are more, let's say steady state, almost like steady state experiments. I mean, they are looking at relatively long stimulations, and they don't have the time resolution. Although, of course, there are people in especially in animal models, where they do line scans and they have very, high temporal resolution across the layers. I don't have, my memory about those studies is not so good at this point to somehow tie those studies to your question about this propagation effects.
They're looking at onset. So those study, Xin Yu, I now collaborate with him at Martinez. So he's doing this line scan. He did it with Alan Koretsky, right? So now he's doing this at MGH. So in this studies, they're looking at onset, which makes sense, right? So onset obviously is gonna, start somewhere. Yeah. Sorry, Ravi, you have your hand up already for a while.
Yeah, I was just going to throw another wrench into the works. So, I think I agree with Camille. It's certainly, in general, and certainly for BOLD, it's the blood flow is dominating, and that's why we, primarily see positive changes. Atina has data that, we'll be submitting in the next month or so, where she's looked at vaso and BOLD across the layers in individual ocular dominance columns. So you can look at the interaction between the columns as a function of layer. So that's a heroic experiment to do. People do columns, people do layers. She's done both. And all I will say at the moment is the conclusion you would draw about the interactions between layers or the interactions between columns depends on the method you use. So your interpretation of what is inhibitory or excitatory, things like that, depend on whether you're using a blood volume technique or whether you're using a blood flow / oxygenation technique. And so, you always, you have to go back to the touchstone of what is the electrophysiology showing? What's the single unit? What are the LFPs doing? What are the vascular units doing at the level you're doing, Anna, layer by layer, to try to figure that out. But I can tell you that certainly if you use the two different techniques that are very popular right now, you would come to very different conclusions about the underlying neuroscience. And so it's very important to understand what is going on so you can actually interpret these. And coming back to the question that I think was asked earlier, we assume certainly that, the mitochondrial density, oxygen consumption, and capillary density are all linked. That's why layer 4 seems to have, the highest capillary density, at least on the assumption that maybe nature engineered that to be the worst case scenario when you have 100 spikes per second kind of thing. And resting state may be that balance is different. My question to you is the CMR02 or the higher CMR2, like that seems to be a disconnect in layer 1 because you don't have a particularly high capillary density there, but is that just made up by the oxygen diffusing but from the arterioles?
Well, but yeah, so first you're right that there is diffusion from arteries, but in terms of kind of cytochrome oxidase mitochondria, you probably just don't need it, So currently we're saying what's higher and what's lower, comparing at the same resting state condition between layers. So my hunch is that when we start looking at, let's say, rapid eye movement sleep, when there's this huge vasodilation, right? Much more than what we normally experience in awake condition. So under these conditions, it may be that we will just discover that there's like a massive load of CMR02 on layer 4. And so currently when we say what's high, what's low, it's all kind of across the layers for the condition of resting state where the mass is sort of spacing out.
But could you, this is speculation. Suppose there was a high increase even during, rapid eye movement in layer 1. Could you provide the oxygen required for that just from the low capillary density, but the lots of these penetrating arterioles? Is that enough to provide the oxygen demand? That's why you don't need a lot of capillaries.
It's a very good question, and it's a quantitative question that requires, doing this experiment and seeing what the numbers are. So currently, it just looks like there's high oxygenation all around, right? Yeah, so as a matter of fact, if we just look at the oxygenation, we see that layer 4 has higher oxygenation at rest than other layers, which is also a very interesting finding. And I stand behind this finding. This already is a third study where we see that. So layer 4 has, to start with, just at resting conditions, higher oxygenation of tissue. And you can think of this as like an extra oxygen buffer. So it's not only that we have lots of mitochondria in there and enough supply, but also we kind of place a little buffer in there of oxygen availability. So when the thing kicks in, we don't know what the thing is, right? But when that type of activity kicks in, that really puts that load on layer 4, we don't drive it into hypoxia. And so the reason mechanistically why there is higher tissue PO2 in layer 4 is simply because the vascular density is high in layer 4. So the vascular density is higher. In addition to this, our measurements show that there is the CMR2 there is lower at rest. But even if just assuming that CMR2 is all the same.
But would you say that a mitochondria in layer 1 and a mitochondria in layer 4 have the same PO2 at the mitochondrial level?
Well, that's going to be dependent on what the demand is. If
In the
you're
resting state.
in the resting state, it looks that the mitochondria in layer 4 have a little more oxygen around.
Okay, which would make sense from what you just said.
Which kind of makes sense if you know, like if you knew exactly what is happening when you're not looking. Okay. So currently we're looking at the resting state. So we're not looking at this metabolically demanding conditions. But when that condition happens, it's going to be super helpful that there is extra oxygen in tissue there.
That's really interesting. We're at about 2.5 hours now. So Amir, if you want to give us our final question, we can wrap up for today.
Yeah, so it's not a question, it's just a follow-up on the very interesting discussion between Anna and Ravi right now. So indeed, it's an amazing phenomenon. There are many synapses in layer 1, and you don't find capillaries. And you know, the hypothesis that arterioles supply the oxygen, hopefully the methods can show that there is oxygen everywhere in layer 1, because arterioles that penetrate cortex are relatively sparse. And if they can really give oxygen everywhere, why do we need the capillaries? So another possibility, another hypothesis that come up to my mind is that there is transfers of vesicles and energy within a cell. Is it possible that synapses in layer 1 get energy from lower parts of the cells that are in layer 2 and 3, etc., etc.?
I think that that's a very nice question for us to ponder over as we head out for today. But I just wanted to very quickly thank all of our speakers very much. We saw some really amazing talks today. And I always really love it when these turn into really nice discussions afterward. That's really what we have in mind for these sessions. So I want to thank everyone for coming today, and we will keep everyone updated for the next layer-fMRI dinner event.
Thanks. Yeah.
Thank you.
Thank